And so I was working with, there was a case of highly resistant salmonella that came into our laboratory, and we did perform susceptibility testing, and lo and behold, it was resistant to ceftriaxone. And we contacted the CDC, and we had some EIS officers come out, and our state epidemiologist went out and contacted the family, and we worked up this case and found that the isolate was linked to an outbreak of salmonellosis in cattle. And what happened is the veterinarian, the way that we thought the case worked out was the veterinarian who was taking care of the cattle gave salmonellosis to his child because the child was taking Pepto-Bismol, et cetera. Anyway, the child came down with salmonella. The child was fine. There were no complications. But what was really interesting is that from my standpoint, from my viewpoint, we were just working up a case, and we were talking about antibiotic resistance, and I didn't really think it was a big deal, because we talk about antibiotic resistance in the hospital all the time. We talk about it, about how we need to decrease our antibiotic usage all the time, and all of a sudden, when this paper came out, that day there was this ground swelling of people that were against me, and I had scientists from UNL call me a scientific hack for this particular paper, and so it was a real interesting time for me. By what I consider just a fairly standard paper. The reason it was published in New England Journal was that it was the first time that this was found. But it was just a real interesting time for me, and it was echoes some of the thoughts that were started at the beginning of this meeting. Is that how there's not a lot of discussion between various groups that work on very similar aspects of public health. So I've learned a lot since then, but again, it was a real instructive time for me, and I was sharing that story with Dr. Raymond earlier. And so as we were having that conversation, he asked me what I wanted to talk about, and I didn't really have a good idea. And since we study Staphylococci, I thought that I would talk about livestock-associated MRSA, and I know it's a big topic right now. And so what I wanted to do is tell you a microbiological story of how population geneticists and population biologists are coming up with ideas about how livestock-associated MRSA is developing and evolving. So it's a very different type of a story than what you've heard so far. And I'm going to get somewhat specific, so I imagine Dr. Raymond might throw something at me, but I'm going to try to get you through all this. And I hope that at the end that you'll understand some of the terms that are thrown around with livestock-associated MRSA or with community-associated MRSA, so we can have a good conversation about this. So I think that I always like to start off with this slide here, and this is a slide from 1941. This is the pre-antibiotic era, looking at the overall mortality rate of staphylococcal bacteremia. And the mortality rate was 82%. And so if you look at the causes of disease there, I think the thing that strikes me is that you have cases of people who are just maybe getting a splinter of some sort from a fence post and coming down with staphylococcal bacteremia and dying. And this happened quite frequently. The only other organism that had a higher mortality rate was Bacterium friedlander, which is Klebsiella pneumonia, with a 100% mortality rate. So we had that era, and then came along the antibiotic era. And I think this is a really interesting story. It was mentioned earlier about the discovery of penicillin. And if any of you ever want to read a real interesting story about a biological discovery, the concept of how Fleming found penicillin, but he didn't have the expertise to actually purify it. And then Ernst and Florey came along and purified it. But because of the Blitz in London, they didn't have the capability of actually making large quantities of penicillin. So they came to the United States, and Big Pharma took it and actually patented the process in the United States. And then so when they went back to London, they actually had to have patent rights back to the United States to make a lot of penicillin. So it's a real interesting story. But once penicillin was put into use, right after that, we started to have penicillin-resistant Staph aureus. Now, if you actually ever have a penicillin-susceptible Staph aureus, penicillin is by far the best antibiotic you can give or you can use against Staph aureus, but most of the time we have resistance against penicillin. To combat penicillin-resistant Staph aureus, pharmaceutical companies made Methicillin, which is a penicillin, but they changed one of the side chains so that it's not penicillin, or the Staph is not penicillin resistant anymore. So Methicillin was used quite a bit during that time period from the 60s up until current. There was also another drug that was made early on, Vancomycin, which is used widely to treat methicillin-resistant Staph aureus. During the 1990s, we had Vancomycin-resistant enterococci, and we were waiting for Vancomycin-resistant Staph aureus to occur. In 1997, we had what's called VISA, or Vancomycin intermediate Staph aureus, and in 2002, we had Vancomycin-resistant Staph aureus. So there's quite a bit of resistance within Staphylococcus aureus that we have to worry about. In our own institution right now at UNMC, we have about 45% of our isolates are resistant to methicillin, and about 95 to 97% are resistant to penicillin. Now, Staph aureus is a highly significant, well-armed pathogen. It has a wide variety of different toxins. It has a wide variety of different resistance mechanisms in the sense that it can resist various activities of the immune system. So if you knock out one particular aspect of virulence factor of Staph aureus, it has about 15 others. So it's a really well-armed pathogen, and it's a very interesting organism to study. It causes a wide variety of different infections, in contrast to some organisms that are more localized to the respiratory tract or something like that. Staph aureus can cause skin and soft tissue infections and cellulitis. It can cause metastatic seeding. What's in the blood can cause osteomyelitis. In this case here, we have a case of endocarditis from metastatic seeding. It can also cause a lot of toxin-mediated diseases. Those of you who are around in the 1980s know and remember toxic shock syndrome toxin. Also staphylococcal scalded skin syndrome. It's also a very significant cause of food poisoning, staphylococcal food poisoning. So it causes a wide variety of different types of diseases, from skin and soft tissue infections to very serious infections such as osteomyelitis, endocarditis, and also toxin-mediated diseases. So right now, it's the most common cause of endocarditis, most common cause of nosocomial or hospital-associated infections, most common cause of skin and soft tissue infections, cellulitis, osteomyelitis, septic arthritis, and it's also a very common cause of bacteremia, nosocomial pneumonia, foodborne disease, etc. So it's a very significant pathogen, especially in the hospital environment. Okay. So here's where what I want to do is talk about the livestock-associated Mrsa, or LA-MRSA, but I think to tell that story effectively, what I'd like to do is start off with telling you about this story that's evolving within the human population of Staph aureus, and this is called community-associated or community-acquired methicillin-resistant Staph aureus, or community-acquired Mrsa. And this, if you happen to see reports in the paper about Staph aureus disease, they'll maybe use the terms of superbug or something along those lines. That's because those particular isolates are resistant to methicillin, and they're causing in the past, where we thought that they were mostly in the hospital. Since about 1999-2000, these organisms have now found their way out into the community. So they're causing infections in locker rooms, in patients who have no access or any type of association with the hospital environment. So it's a very different situation than what we've seen in the past. So methicillin-resistant Staph aureus, or Mrsa, one thing that you need to know is that methicillin is no longer an antibiotic that we use against Staphylococci. It was the first one that was made, but because of its stability, nobody uses methicillin as an antibiotic anymore. And the antibiotics that are very similar to that are called oxacillin or nafcillin. So the CDC a while back tried to change it from Mrsa to Orsa, Oxacillin-Resistant Staph aureus, but it didn't hold. So people still call it Mrsa, even though we don't use methicillin resistance anymore, or we don't use methicillin anymore. If an organism is resistant to methicillin, it's also resistant to all cephalosporins, all penicillins, and the carbapenems. So it's essentially resistant to all beta-lactam antibiotics, and that is a very significant part of our armamentum against bacteria. So, before I continue on with this story, there are 3 big points that I want you to know about. First is that beta-lactam antibiotics, the way that they work is that they bind to enzymes in the cell wall of bacteria, and they don't allow the bacteria to actually build that cell wall anymore. So once the antibiotic binds to these proteins, then the cell stops growing and it'll burst. Okay, so it binds to these to these proteins in the cell wall. Almost all of Staph aureus are resistant to penicillin. So penicillin was that first antibiotic that was made by Fleming, and almost all Staph aureus are resistant to penicillin because they make a particular enzyme that's called a Beta-Lactamase, which cleaves or destroys penicillin. Okay, so almost all Staph aureus have this enzyme now, and it's found on a plasmid. Dr. Tox was talking about plasmids and Salmonella. Now, that particular penicillinase cannot hydrolyze methicillin or oxacillin. Now, the way that you become resistant to oxacillin or methicillin is that you've acquired a new gene. And this new gene is called MECA. And so maybe some of you have heard of this gene called MECA, conferring resistance to methicillin. And MECA is the gene which confers resistance. Now, MECA is found on this element in the genome of Staph aureus that's called SCC-MEC, and there are many different types of SCC-MEC elements. And it's being transferred around by a bacteriophage. Now, the point here to note is not how this works or what have you, but methicillin resistance is transferable. We were talking about earlier about the CMY2 gene from Salmonella and how it's transferred from different Salmonella strains to other strains on a plasmid. This is a little bit of a different mechanism, but Staph aureus has the ability, through actually a bacterial virus, to mobilize or move methicillin resistance from one Staph aureus to another. And that's an important point to note and to remember about how MRSA evolves in livestock animals and as well as in humans. Okay, so what does this slide mean? This slide is important for discussion of MRSA because when we start talking about strains in MRSA, you may hear the words USA 300 or ST 398. All that is a way of population biologists or geneticists of defining particular strains. And the way you do that is you take the DNA of the organism and you sequence certain parts of it. And from those data, you can then say, yeah, this particular Staph aureus strain is part of this particular, they call it clonal complex or sequence type. And these circles up here are the way that it's represented. So those particular circles, all the strains within that circle are highly related strains. Okay? And so those particular strains are given names. And so when people talk about livestock-associated MRSA, they give these particular strain names associated with that. So it's important to note. Okay. Now, in 1999, we had we thought we had a pretty good idea of what MRSA was, what type of strains encompassed Methicillin-Resistant Staph aureus, and worldwide, MRSA was found only in the hospital. There are very few instances where somebody would come into the emergency department and the physician treating that particular patient would have to worry about an MRSA infection in those particular patients. All of a sudden, we started to hear about patients that were from Native American communities, Native Alaskans, that had these very high rates, and this was discussed earlier, very high rates of carriage of methicillin-resistant Staph aureus, and these particular patients had no association with healthcare facilities. So it wasn't like a situation where they had their mother or father worked at a hospital and they transferred that particular organism back to their child at home. But instead, we had these particular kids that had very serious infections. And in this particular article from MMWR, these particular children had necrotizing pneumonia and actually died from methicillin-resistant Staph aureus infections. This was very different than what we had seen before. Again, most of these isolates were found only in the hospital. These are the types of infections that we were seeing at that particular time, and also since that time, since 1999. Most of the time, we're seeing a lot of boils, pustules, skin and soft tissue infections associated with these patients that are coming in to the ED. However, we've also had some very highly publicized reports of very serious pneumonia with patients with community-acquired MRSA. Patients who have necrotizing fasciitis, which is a very deep-seated infection that normally associated with group A strep, with patients with community-acquired MRSA. Very somewhat unique infection associated with Staph aureus, which is pyomyositis, that we didn't necessarily see before. So we had this changing of the resistance from normal susceptible Staph aureus in the community. All of a sudden now these organisms are coming, or these patients are coming in with highly resistant, methicillin-resistant Staph aureus infections. This particular paper was published in 2007, and essentially what this paper did was looked at the prevalence of community-acquired MRSA within emergency departments within the United States. And the really shocking thing associated with this paper was that if they extrapolated to the population of the United States, that they estimated 94,360 infections with community-acquired MRSA in the U.S. And 18,650 deaths associated with it. The number that was somewhat striking is that this was more deaths than is associated with HIV/AIDS right now in the United States. Okay, so at that time, when we started looking at this in 1999 and 2000, we actually had a Native American population that we studied in Nebraska, and the question was this a hospital strain that we were well familiar with that had escaped the hospital and was now found its way into community? Or was it a normal strain that we find in the community that had acquired the MEK-A gene? Remember I said that the MEK-A gene that confers methicillin resistance is transferable within bacteria. So through a lot of work, and the CDC did a lot of this work, there was a particular strain that is called, with this arrow here, which is called USA 300, which is the major community-acquired MRSA strain in the United States. This particular strain is not restricted to the community anymore. When the outbreak or the epidemic first started, USA 300 was primarily in the community, but now it's found its way into hospitals. It's also been isolated from companion animals, where you've had patients that are colonized with USA 300, or have had infections with USA 300 that have transferred those to companion animals. There have been situations, there was a report where a worker at a zoo had a USA 300 infection and was taking care of an elephant and actually infected the elephant with USA 300. So it's clear that there are the USA 300 has the capability of not only infecting humans but also infecting other animals. So what's so special about USA 300? Now this is where I want to spend just a little bit of time with this because I think this is important for the discussion of livestock-associated MRSA. So we don't really know what is really important about USA 300. We have some really interesting hypotheses about what's special about USA 300 or the community-acquired MRSA, which is causing somewhat of an epidemic in the United States. If you look at these particular facets or factors that are found in USA 300, we know that there are particular virulence factors that are found in USA 300 that are not found in normal Staph aureus strains. One of them is called Panton-Valentine leukocidin. And this particular toxin may be very important for disease in pneumonia. We also know that these particular strains actually express more of a toxin called alpha toxin. And this may be very important for patients with pneumonia. But one of the more important things is that these particular strains have this new gene that is called it's called a genomic island, which is called the Acme island. And this particular island is thought to actually confer an increased ability of the organism to colonize skin. So in the past, most of Staph aureus primarily colonizes individuals in the nares, but this particular set of genes has the ability it's thought that actually causes increased ability to colonize skin. So therefore, not only can patients be colonized in the nares, but also you can carry it on your skin as well. So therefore, it's much more easy to transfer these particular isolates to other individuals. So although we're really not clear about exactly what's happening here, we do have some really interesting hypotheses what's special about USA 300. And the other thing that we know is that this is not the first time this has happened. We have had waves of particular isolates that have occurred within our population. Now, we've only gone back really to the 50s because that's really where our data set is. But we know that, for instance, there was a particular group of Staph aureus which are called phage type 8081, which happened in the 1980s, and since the 1950s. And since that time, we've had multiple waves of Staph aureus that have invaded the United States. And it's really not clear why that happened. So if you can follow up here on the left, top left, you have multiple Staph aureus strains, let's say, in our environment. For some particular reason, one particular strain has some particular success. Let's say we use our USA 300 example, and that particular strain has the increased ability to colonize skin. So let's say that's the particular trait that allows it to succeed. Then that particular clone or strain can expand, colonize other individuals, and then over time, you would think that based upon herd immunity or other particular factors, that particular strain would then be kicked out and another strain would then take its place. And although that hasn't happened yet, USA 300, we think, has been around for about 15 years or so as the major predominant Staph aureus strain in the community. I predict that over time, USA 300 is going to be kicked out and another strain is going to take its place. So how is that important for livestock-associated MRSA? First, in contrast to some organisms, let's say, for instance, Streptococcus pneumonia, or group A strep, those particular organisms, or Shigella. They generally infect humans. Other organisms, such as Salmonella, Campylobacter, and Staph aureus, has the ability to infect multiple species, including food animals. So we know that Staph aureus can infect and cause disease in cows, sheep, goats, poultry, rabbits. And if you look at those particular strains that infect those different types of animals, most of them are host-specific. So if we did that specialized population biology analysis of those strains that infect rabbits, let's say, versus those strains which cause humans, we could pretty much say that those particular strains are very different from one another. However, some strains have evolved or adapted to be able to colonize multiple animal species. Some of these strain names that remember how I said that these, using population biology, we can call these strains these particular numbers. Some of these strains are called like ST1, CC5, ST8, and a real famous one that's called ST398. So we know that these particular strains can cause disease both in humans and in animals. And I'm going to give you two examples here when I wrap this up. The first example is this particular strain, which is called ST398. I think this is the strain when they started talking about livestock-associated MRSA, they were talking about ST398. And this particular strain really started, I think, the whole research enterprise associated with MRSA and food animals. This particular strain is widespread in pigs in the Netherlands, Europe, Asia, and the United States. Unfortunately, it has the additional capability to colonize and cause disease in humans as well. But in contrast to other strains, once people have an infection or are colonized with ST398, they really don't transfer it to other people. So if you take a look at people who pig farmers, and if the pigs actually have ST398 Staph aureus, typically there is an increased risk for infection with ST398 in pig farmers, and there's an increased risk for colonization in the nares with ST398 MRSA. However, they typically do not transfer it to other members of the family. How did ST398 evolve? So I think that there's a lot of questions within circles that study this. Did this particular strain come from humans? Did it come from animals? And there's been a lot of recent study on ST398. And what's happened, the geneticists have found that the original ST398 actually colonized pigs from humans. So we have found the original ST398 in human populations, and it's thought that it adapted to swine. While it was in pigs, it picked up this particular element, which allowed it to be resistant to methicillin. It also picked up other resistance elements to tetracycline, macrolides, and apomycin. Also, we found that once we have found that strains that reinfect humans from pigs have changed a little bit, so that when you analyze those strains that are infecting humans, they've actually picked up other human-specific virulence factors. So the story with ST398 is that it originally was in humans and was transferred into pigs. Once it was in pigs, it picked up particular specific virulence factors associated with causing disease in swine. And now it's being transferred back into the human population. And once it's in the human population, now it's picking up extra virulence factors that allow it to cause disease in humans. So you can see how the propensity of Staph aureus to acquire new genes is allowing it to cause disease both in humans and in animals, and also to mediate antibiotic resistance both in humans and in animals. One other example. This is a strain that's called CC5. And this Staph aureus in poultry is a major cause of lameness due to osteomyelitis. The majority of, when you look at those particular strains which cause disease in poultry, they belong to this one strain called CC5. This particular strain is also found in the human population. Population biology studies have suggested that this particular strain originated in Poland. And about 40 years ago, around 1970, there was a jump from humans, a human CC5 strain, into poultry. And now that particular strain in poultry has picked up poultry-specific virulence factors. It's really interesting. So an example is that they've picked up an avian-specific cysteine protease, which mediates virulence. Another really interesting thing is that this particular virulence factor in humans is called protein A. And this particular protein binds to antibodies. And since chicken antibodies and human antibodies are very different, that particular gene has been lost in the population. So since it doesn't need that particular gene anymore in poultry, it's been lost in that population. So you can see how it's being evolved and changing within the poultry population. And we've had global dissemination of that particular clone. So I think that the conclusions here, when we're studying Staph aureus and we're looking at how Staph aureus causes disease in animals, is that Staph aureus is highly adaptive, and it can evolve very quickly due to the acquisition of genes from other sources. So it can pick up virulence genes, and it can pick up antibiotic resistance genes. And we know that we can have situations where animals can transfer MRSA and Staph aureus to humans, and we also have examples where humans have given Staph aureus to food animals. And so I think that the thing that we really need to look at is what particular selective environments are within those populations, or within those environments, which are allowing this particular selection to occur. And I know that antibiotics on farms probably have something to do with selection of some of these pathogens within food animals, but I don't think that's the whole story. I think that there's probably some other selective pressures that are in play here. For instance, we know that in certain populations that never take antibiotics, such as people on specific islands, Pacific Islanders, native Alaskans, never see antibiotics, and they have an extremely high rate of MRSA carriage. So there's got to be some other particular factors which are causing some of these resistance factors to stay within particular, or be kept within particular bacteria. In Staph aureus, USA 300, it may be that the gene which confers or allows the organism to colonize skin may be one of the major selective pressures within that particular population. And we know that there's a genetic linkage between MEK-A and that particular gene as well, so they may be kept together. So I think there are a lot of really interesting biological factors in play, and I think that antibiotics and use of antibiotics is clearly a significant part of that, but I think that we also need to think of other selective pressures as well.